Good evening, guys, and welcome back to the first episode of tonight's channel. How's everybody doing? My name is Patrick. I see some familiar names in here, which I'm always excited to see on this lovely Sunday afternoon. I also have my other channel expert with me, Eli, who will be manning the Q&A over there in the right. So any questions that you guys have throughout tonight's channel session, please be sure to fire away. I see some familiar names in here, so I want to say what's up. A lot of you guys comment on MCAT channel here, and of course a couple of people from my class. So good to see you guys for one of my favorite topics to cover on the channel, and that's going to be fatty acids and hormones in metabolism. Now, who at first glance of this topic kind of shuddered and went, ooh, this might be a tough channel session. I'm not really a huge fan of this content so far, and I do not know it as well as maybe as I should have. Yeah, okay. So in all honesty, there's a lot of material that we're going to cover in tonight's channel session. But what's most important about this is the big time learning strategy that we're going to cover on how to conquer through a lot of material that at first glance just seems like a lot of memorization. So what we always highly encourage with any of our 3 metabolism sessions is to make sure that as you're going through, make out a question list or a topic list of the things you need to go back to and really just commit to memory. One of the things that we're really going to be focusing on throughout tonight's both learning goals is going to be how can we use this information to apply it directly back to the MCAT. So even though we'll be going through some of the mechanisms a little bit quicker if you're not as familiar with the terminology, the enzymes, or the nomenclature, you'll notice that it's huge opportunities for wow moments to get more questions correct in kind of a limited time. Because remember, what are we really here for? Points. Points is the name of the game on test day, and let's take a look at how fatty acids and hormones can get us a few more points to bring up that MCAT score. So in tonight's episode, what we're going to learn about is we're going to identify some of the key enzymes in fatty acid synthesis and oxidation, specifically focusing on certain parts of the mechanisms that we just need to know to understand big picture concepts. Then after that, we're going to talk about the different energy states and functions of major hormones in the energy balance. I always like to think of this as food. Sometimes we hear about these fad diets out there, like the Atkins diet, where you don't eat any carbohydrates. What it's kind of doing is, they'll talk about a lot about this in marketing, of activating alternate energy systems. You guys all heard about this, right? I don't eat any carbs so I can go on my beach body spring break tour, right? I think if you can kind of start to understand that, it kind of always gives me an idea in the back of my head of, okay, we're alternating different energy pathways. How does it all play into the big picture? Because in the end, we still want to end up with the mitochondria producing ATP. So in learning goal number 1, we're going to talk about those key enzymes in fatty acid and ketone balance. So where in the past did we talk about pyruvate and acetyl coenzyme A? For those of you guys who've seen it either in the channel session or in your review. I really want to make sure we tie this into big picture, right, guys? It's all about those points. Exactly. We talked about the citric acid cycle and specifically glycolysis. So in order to make that pyruvate, what was the precursor that we needed to do, or we needed our body to consume in order to go through or activate that pathway? Glucose. Excellent. So you guys already got the big picture here. Now, what we say is, can there be certain times where maybe we don't have as much glucose readily available to us, and then we can switch or alternate pathways to use our fatty acids and bring it back to that acetyl coenzyme A and the citric acid cycle. So if this is some of your first channel sessions and you haven't seen that Metabolism 1, that's where we cover that citric acid cycle. But think big picture. How can we relate this fatty acid back into so we can get that alternate energy pathway? All right. Fatty acids are primarily broken down to ketone bodies and acetyl-CoAs. So some tissues within our body actually almost entirely rely completely on this metabolism to generate energy. What's one of the types of muscles we usually talk about in physiology that predominantly requires this pathway? Anybody happen to remember? It can be a nice, quick, easy point on test day if you noticed it. Remember, we want to try to take shortcuts. Exactly, guys. Cardiac muscle. Excellent job. So we're going to tie this back in, or most importantly, I want you guys to take away from this lesson is, even if we're going through these pathways and these big diagrams and charts and you kind of get lost, I really want to make sure we're covering the most high yield information that you guys can apply readily back to questions. Okay? So let's check this out. Now, our fatty acid metabolism, not metabolism, excuse me, mostly this biosynthesis is going to happen in the liver. But what we're going to show you guys here is a pathway and how it all relates back. Oftentimes when I first learned this, I would get super confused as to the precursor and then what that ultimately led to. Does anybody feel like once you look at this, you're like, wait, what goes to what, right? That's usually one of the most common complaints we get from students is like, I know glycolysis. That was usually the big thing that we had to bring back, right, within biochemistry, and usually went through the enzymes, and you went through that whole process. What we want to understand in the fatty acid metabolism is how does this plug back in to still go through that second half of our cellular respiration like we talked about in metabolism 1, but how did we get there, or how did that process form? So you'll notice that we're talking about the cytoplasm at first, and acetyl-CoA is really going to be that first part that we're trying to focus on. So what ends up happening is you want to get that fatty acid palmitate. Now, what is this in the overall picture of fatty acid metabolism? Why do we want to produce this is the question. Anyone happen to remember? Exactly. So if we start thinking storage, Esther, I really like that answer, then that starts to give me a clue. But here's the big picture for those of you guys who are not as familiar with this information. Now, some of you guys might have went and learned ahead, which is fantastic. What is our ultimate goal once we take this acetyl-CoA and convert it into the fatty acid palmitate? What's the main goal, the main takeaway here? Why do we want to do this? Where are we going with it eventually? Okay, to make ATP. What is ATP? This is why I want to kind of do that quick refresher. I'm only going to do this for the first part of the session so we understand where this comes from. Exactly, our energy. That's why we literally say alternative energy pathways. This material is not as difficult as it initially appears to be. However, what we need to understand is acetyl-CoA, wait a second. Now I see acetyl coenzyme A being present in both the mitochondria and the cytoplasm. What's up with this? Well, remember, if we have certain intermediates here, we have to understand where those intermediates are from, okay? Most acetyl-CoA is going to be in the mitochondria, okay? But here's the problem. The mitochondria is impermeable to acetyl-CoA, which is going to bring a transportation part, which is really going to be a big clue here. So what happens is once in the cytoplasm, the acetyl can be reformed from citrate using ATP, you can actually get a process of transferring out of the mitochondria. So this OAA, excuse me, I'm bringing this acetyl coenzyme A, even the citrate, now I have permeability across that membrane. Remember, if it's impermeable, I really have nothing to be able to bring through here. Now, once that happens, I'm using that ATP to convert back to my acetyl-CoA. So here's the question, and if any of you guys have seen me teach Metabolism 1, I always use the idea of the bank. Using the bank, we either have to deposit or we're withdrawing energy to use later. What are we doing here? Are we depositing energy or withdrawing energy? It sounds like a super weird analogy, but I'm telling you, it works when you think about it later on. Are we depositing energy or withdrawing? Excellent. So you guys are catching on to this nicely. You're withdrawing energy from the bank because ATP is costing energy. You're using up or using that money in order to convert to acetyl coenzyme A. So what happens now, does anybody remember what OAA is going to be converted into? Remember that NADPH like we talked about in other pathways or electron carriers. Anybody happen to remember where that's converted back into? Because we don't want it to just sit there in the cytoplasm, right? So there's got to be a cyclic process. Remember, it's going to bring back to malate. Okay? Now, this is where usually the confusion becomes, because remember, we're in a completely different process here. This is cyclic, so there is no arbitrary start and stop point. What we want to understand is where we are at in this ideal process. So if you have acetyl coenzyme A brought outside into the cytoplasm now, the OAA can be produced and converted to malate and form malate to pyruvate eventually to bring back in. But what we need, and remember, oftentimes our enzymes are just named according to what they're going to act upon, is the malic enzyme. So once that happens, can I bring pyruvate then back into the mitochondria? You betcha. Exactly. So bringing that pyruvate back in, then pyruvate carboxylase and the OAA, we can go back through this cyclic process. But we're not completely done with this, okay? What ends up happening is pyruvate can enter the mitochondria, be converted to OAA through pyruvate carboxylase or acetyl coenzyme A. It's our pyruvate dehydrogenase, which can then be converted to citrate and sent to the cytoplasm for more of this fatty acid synthesis. So the picture or the clock-like pathway we just brought up, notice it was almost clockwise, is really just a cyclic process. Does that make sense, big picture, guys? This is what I want to start with because we're going to start to get into a little bit more of the details, but I want you guys to remember big picture. Remember, the MCAT is still going to be a critical thinking test. We never want to be able to go through this material just for sake of going through the material. What we really want to see here is making sure that we're understanding how this fits in across cross topics, okay? Yeah, that's our pyruvate dehydrogenase, okay? So now, once that happens, I got to think to myself, okay, if I go through this process and I see over here on the right, what's going on in this part to eventually get me that storage with my fatty acids? So the biosynthesis part we have to think about is I have acetyl-CoA. What's going to happen is I need that ATP and that CO2, and I'm going to bring that in to bring malonyl CoA, okay? Now, what I used to do for this, and I think is probably the best from a strategy perspective, is actually make your own flashcards, okay? It's something Eli and I talk about quite a bit, other MCAT teachers, and one of the things that we found is usually the shortest way to get a lot of this wording down is actually make your own flashcards when you're going through and reviewing these either slides when you guys look back on the archive sessions or going back through your review notes. But exactly like Eli just said, you can kind of control exactly what it says, and just the process of going through and actually making those flashcards is something that I think is super underutilized, especially in the process of the MCAT. Because think, does knowing any of this information matter if we don't get any questions correct? Does it really matter going through and just blindly memorizing anything? No, absolutely not. This has to still be able to fit in to answering more MCAT questions correct. Good. So I need biotin acting as a cofactor with my acetyl-CoA carboxylase. All right. Now, once that happens and we get to this, I always like to say, if you ever look back in any of our metabolism decks, okay, what are the things in the pink circles? So this is a quick tip that's really going to help if you look back at them. Exactly, Molly. Good. So some of you guys have been to the other metabolism sessions, right? Anytime you see that thing in pink, remember that's the enzyme that's acting on. So what we always like to think about is if you know the input and you know the output, you can start to piece this together to understand the overall pathway. So what we're saying here is acetyl-CoA is first carboxylated via this endergonic reaction. Excuse me. But what, if you guys remember, exactly, Esther, very good. Blue is going to be the cofactor. You're one step ahead of me, which is perfect. This is our rate-limiting step, okay? So most people forget this part, but what it's doing is the acetyl-CoA is almost literally catalyzing this reaction with the biotin as a cofactor. If I don't get into this, what's the major enzyme that's going to bring me to my palmitic acid? Does anyone happen to remember? The major enzyme. It's like one of the most common enzymes that people talk about, especially, yes, Molly, you got it. First gold star for the day, FAS, right? That's my fatty acid synthetase, okay? So fatty acid synthetase oftentimes will actually just be abbreviated to FAS, is your next key enzyme. But think, if this is my rate limiting step, if I don't have enough biotin or my acetyl-CoA carboxylase, am I ever even going to get to that step to make my palmitic acid? Not going to happen. But why is this important in the overall grand scheme of things? Think back to your MCAT. Your MCAT is a passage-based test. So oftentimes, even in those passages, what do they tend to do? What do they tend to manipulate that usually makes the material, at least upon first glance, more confusing? So think, big picture here. Yeah, the vocab, and then what, Tom, what else can they do from a positive and negative feedback perspective? Because remember, if the MCAT's focused, and especially with the test changes in MCAT 2015, your critical thinking, how can this fit back into the MCAT overall? It's got to be, okay, if you have excess of something or you don't have enough, then you start to get blocks and pathways. So ultimately what we have to think here is you still have to think back to metabolism 1 when we talked about glycolysis all the way into the citric acid cycle and how this is going to piece back together. Exactly. So take what Eli is saying here. You got to watch out for your product and reactants and see what's really being used. But ultimately, the malonyl-CoA can be incorporated into a fatty acid via this reduction and decarboxylation by your fatty acid synthetase. So what happens oftentimes in your biochemistry classes, which isn't as important when you're going through and studying, is you go through huge biofeedback pathways according to what's being made with acetyl-CoA, and then ultimately the malonyl-CoA, and then finally bringing that acetyl-CoA back through. So this NADPH is going to cost 2 times, but what we need to understand is how can we make a 2-carbon acetyl group into a how many carbon chain? So a lot of you guys were throwing this in the chat earlier, which is what I'd love to see. I could tell you guys were doing your homework here. So upon first glance, it's going to be that 16-carbon chain, okay? So what happens is you get this huge pathway, but you need to understand, okay, what are these arrows and what is this major thing in acetyl groups? What is that doing, or what is that starting to carry? Notice how we go from one step to the next here. What is it carrying for us? Yeah, we're adding our carbons, right? So you understand that if we have NADPH, what is that really the source of? Why do we have the NADPH there? Exactly, right? You need that source of electrons, which is going to be kind of your energy pool, to be able to bring this back through. So if you have your acetyl-CoA and you have acetyl-ACP, what I need to understand then is if I don't have enough of any of these cofactors, enzymes, or inputs or outputs, I'm stuck. But ultimately what we're talking about here is we're making this 2-carbon acetyl group into a 16-carbon chain, okay? So when we first convert to the malonyl-CoA through carboxylation, we're bringing this all the way through because the malonyl group is also the link to ACP before, right here, box it off, the reaction begins. But this is the part that's just memorization. You got to understand the inputs and what's being required to ultimately get the output. So what happens here is if you don't have this first part, again, what's going to happen? Think big picture. If you don't have this first part that I just brought in red, can you really get that biosynthesis or your fatty acid? Exactly, Grant. Good. You have that blocked pathway. So what I want you guys to take away, if nothing else at the end of this session, you might have to go back and review a lot of this nomenclature, because in 55 minutes, if you are familiar with the nomenclature, it might be a little tough, but I want you guys to look for the arrows. If you guys can start to see this process, if you have, just as Eli's put in the chat, those rate limiting steps, if you can't go through that step, nothing else is going to happen. So you have to go through an alternate pathway or metabolism. That's why you hear people talk about this so often. So what happens is you can start to build. Notice, what do I continue to start to add here? That's the only thing that I'm adding. My hydrocarbons. So what you're doing is you're literally starting to build this as you go through. So when I circle this over here on the right, I can change my marker color. What really is all this stuff over here to the right that I just circled in green? What is that? I think big picture again. That's just my byproducts, right? But theoretically, can't I recycle those byproducts to bring them back in to just continue to add more and more carbon to this chain? Yes, sir, Tom. I love the enthusiasm. This is really what it's all about, right? It's almost like you can think of metabolism. And I know that I was using that bank analogy of withdrawing and using it, but think about it. If you guys were factory owners and you had to make, I don't know, sneakers or products or whatever, or maybe you were printing MCAT books, who knows? Don't you have to spend a little bit of money to make your product and sell it for a little bit more? Sounds like a really wacky analogy, but I had this crazy biochem professor that even had like the mad crazy hair, and it always clicked in my head. Because if you think, if I have to withdraw some of that money or cash or energy, and then it's ultimately going to allow me to produce or store more energy later, is it worth the process from an alternative metabolism pathway? You betcha, absolutely. Because think, if you're going through, which we're going to see in learning goal number 2, let's say, for example, and this one of the most common studies that we always cite with fatty acid metabolism and humans being able to live off of just fats is in like the North Pole. If you start to run out of a glucose source, can we still survive for at least an extended period of time off of fats and proteins? Yeah, absolutely we can. That's the whole idea of this process. Still think big picture in the long run. So as soon as we get to that carbon, now we've made palmitate. And like you guys correct saying earlier, that's that 16 carbon source. Exactly, Esther. I'm so big on that. The more examples that we can think of in the big picture, then it's going to start to make sense why we're memorizing these nitty-gritty details. Because in the end, all right, the more hibernated, the better, right? In the end, what we really want to do is be able to apply the big picture concept back to the MCAT for the right reasons. If you miss that part, then how many of you guys have ever taken a class where you literally just start to memorize for memorizing's sake? Anybody ever done that before? It's like you get so far into organic chemistry or anatomy, and you're just rote memorizing. You're like, why on earth am I doing this, right? But that's not what we're encouraging here with our metabolism lectures. Exactly, I hate it too. It's super annoying, and at the end of the day, you don't really feel like you learned anything. But if we start to understand the big picture of why we're storing this all up, we have stored energy to be able to call on later. All right? Now, bringing this back together, I want to go back to our big picture. So we said, why did we put that big circle right there? What was the point of showing that process? What was the point of showing that process? The cyclic, exactly. But now what we can see here is when we had that melanoid-CoA, what was the enzyme that I want to put right here to be able to convert into my 16 carbon source? What was that enzyme? Boom, Tom, Grant, excellent. Now see, all of us are remembering why we actually have to know that enzyme within that source. Excellent, guys, very good. So what we do need, and this is why we're bringing that NADPH back out. Oh, wait a second. Does this make sense now? I want to put a big gold star here, and I want you guys to think about this. Why on earth would we draw it that way if we could have drawn it another way? Because there's a method of the madness. Notice this NADPH here. Didn't we just look on the previous slide? Didn't I need 2 NADPH? Yes, Rochelle, I could not agree more. This is why I love to be a biochem nerd. Because at the end of the day, we don't really understand biology until we understand these pathways. When you're connecting something like this malic enzyme, and producing that NADPH, then you can use that in conjunction with that fatty acid synthetase to bring in palmitate. Okay? So, big picture, just in the first 20 minutes here, to make this a little bit less intimidating, what's my major takeaway? Why did we show, or why did we introduce this first? Think why. I'm not going to let you guys lead the channel session until you think why. Why? Always question, always challenge. That's what the MCAT's all about. What was the point? I see a lot of you guys typing. Fire away. No big deal. What was the point of showing this first? Don't be shy now. Exactly, Molly. Good. Exactly. Good, guys. All right. If I got you through anything in the first 20 minutes, we see that alternative source of energy. So the thing that we want to do then when we leave this session is obviously go back and make sure we understand what are all the intermediates, okay, cofactors, enzymes, and the like. But ultimately what we're doing is we're understanding then this oxidation and where certain things are going to come from. So what we want to look at now is if we have these fatty acids freely floating through, you can use oxidation of fatty acids is used by most cells by the acetocoenzyme A production. But what you guys are going to see here now, does anybody happen to remember the limiter with the transporter? What transporter always comes to mind? It's always tested under biochem tests. What was the transporter from intermembrane space to the mitochondrial matrix? You'll see it pop up right on this slide. Exactly. Ding, ding, ding. So it's your carnitine transport. All right, really close, Andrew. So check this out. If you have your capillaries, obviously your blood source, your plasma freely floating, and we get this fatty acid. What I start to do is I can bring, okay, the fatty acids taken up from the blood, and it must be transported to the mitochondria from that cytoplasm. So in the cytoplasm here, what I'm starring, are we technically in the cell at that point? Remember, we always want to make sure we're linking things back to our cell physio. We're in the cell at that point, right? Good. We're just not in the mitochondria. So even if you go back to that really simple analogy of the mitochondria is like the power plant of the cell, right? It still is. We just need to be able to bring that fatty acid through the cytoplasm to the factory to bring it into the intermembrane space. Now, are we withdrawing or are we depositing if I'm using ATP here? Are we withdrawing or are we depositing? Think. Am I using ATP? Am I using ATP? Because in the intermembrane space, the fatty acid is activated by this linkage to this coenzyme A. Yes, so it's okay, Tom. I just like to use that analogy because you remember, if I'm taking ATP away from something, I'm using it. So I'm withdrawing it from my source of energy. When that happens, now we get the activation, which is key. And this is where it starts to get exciting. I know biochemistry is not as exciting as everything, but it does. It gets exciting because now we start to see the connection again. So a lot of you guys started to put this in. If I can bring this fatty acid with the CoA and then get rid of it, now I bind it to my carnitine. So what happens there is this is a special carnitine transporter is used to bring it across my intermembrane space into the mitochondrial matrix. Okay, so what happens here, and you'll notice, I want you guys to see a big box. I'm going to highlight it in yellow here. This is literally almost going to be the same stuff. Watch. When I do that, what's the only reason then of binding it if I have bringing it across? What was the only reason for binding it to my carnitine? Why do I do that? It doesn't really have anything to do with the overall path. Exactly, it's just for transport. It's just an intermediate. Andrew, exactly. I need it to travel. Because now, once I bring it back into the mitochondrial matrix, what do I need to do to convert it to acetyl-CoA once again? What do I need to do? How do I do that? What process? Is it oxidation or reduction? It's just a quick test just to make sure we remember our chemistry here, right? So what's going to happen is you have to go through the process of your beta oxidation. So this is really the key here, right? If you look at it, and I know I'm drawing a couple things here. Let me erase this just to clarify one more time. What I'm trying to take away from this is if I see at first, I take that ATP and I convert it with the CoA, and then I bring it to a cross with the transporter, the most important step is really at the end here. I want to see beta oxidation and bring it to acetyl coenzyme A. Now wait a second. If I show that, or I think back to the original part, why did I show acetyl coenzyme A? Let's go back to the first slide of the deck. Why did we show this earlier? What are we doing with acetyl coenzyme A? Aren't we just really looking at the first part of the right side of my alternative pathway? Right? So in the end, I'm bringing this right back to the citric acid cycle. And back in Metabolism 1, we talked about the citric acid cycle, still for the overall production of ATP. Right? Excellent, guys. Good. Right. We want to be making that energy. So let's bring this in. Now, if I have that, ooh, where did I just see this? This is my 16 carbon source, right? So what I'm going to have to do is eventually go through this process, right? If you have this long fatty acid chain, it's going to be oxidized by your acetyl groups and then incorporated into the acetyl-CoA. Beta oxidation gets its name because of the series of reactions that will oxidize the beta carbon, so that carbon 1 and 2 will be broken from the chain. Now what starts to happen is you get that acetyl coenzyme A equivalent, right? Now what we want to do with that next step, remember with your basic organic chemistry, sometimes we learn it in Gen Chem in our classes in undergraduate, but most of the time, remember you guys talking about this in Orgo? Well, think, why do you have to know chemistry before you get into biochemistry? This is exactly that reason. Why does it get its name then? Right? Remember how we talk about that back in Orgo? It's like, all right, well, why am I learning about alpha and beta carbons? So it's like 1 or 2 away. Well, think, yeah, Maui, exactly. Location, location, location. And if you think about it, if you start to understand more, what do you have to do less of on the back end? Which is actually really exciting. It gives you a lot of confidence. But you do less, exactly. It's less memorization in the end. So when you start to put these things together, it's less memorization, which means less studying. So what I start to do then is if I bring in this FAD, and then you convert it to that FADH2, this beta oxidation really becomes the key. All right, you're going to see that the first oxidation just forms a double bond between the alpha and the beta carbon, and then we use our water. Okay, so now what it does is it starts to set me up. I still need to use NAD plus to convert to NADH, but ultimately now I start to get this double bond. Okay, now exactly like Eli's mentioning, we've seen this before. Okay, so the first 2 carbons ultimately have been separated because what are we going to add in my process next, right here? What are we going to add? A lot of you guys have seen this before at one point or another. Think, if I want to split these 2 apart, what am I going to add here? Just one more time. Boom. See this? Little MCAT magic for our Sunday afternoon, and we get it one more time. Now I've split these 2 apart. So really, in the overall scheme of things, what do I need to keep doing now if I have all of these over here? What am I really going to be doing? Just adding coenzyme A. So if you can always go back and ask yourself why, or why am I going through this, that's really the main takeaway. So you get that acetyl-CoA one more time. So theoretically then, if you think back big picture, don't you get a little bit more energy from fats than you do carbohydrates? You're going to get more energy from fats than you would carbohydrates. Yeah, absolutely you would, right? This is why I always say per gram of fats, you have more calories in the long run. All right? Yeah, that's why I'm eating ice cream, right? Well, that big juicy porterhouse. But that's exactly right, because they're highly reduced. Excellent. So again, if we can think back big picture, we start to understand. Now, if you see my next slide here, we kind of go through it. What happens? Yeah, of course, we want to burn more through it too. But this is why when people alternate their diets, you can actually start to see it's actually a source of a little bit more energy. All right? Yeah, we just sit all day, right? Now, you go through that process again and again and again. What we're going to do is continue to produce it. So what ultimately happens here, if you're in that mitochondrial matrix, the rate limiting step, remember, anytime we put brackets around something, what is that? What's the bracket part? Concentration. So the higher the concentration of fatty acids I have in the blood, the more we're going to go through that pathway. But ultimately, at the end of the day, what we're starting to understand, beta oxidation is literally just the conversion of fatty acids to acetyl coenzyme A, right? The rate limiting factor here is what's happening is just going through. If we have enough presence of that fatty acid, we'll start to turn on that pathway, okay? Now, that fatty acid biosynthesis is something that you have to think about in the long run. Because what is going to have that negative feedback system that we looked at on our previous slide from this process right here on the bottom? What did we talk about earlier? If you don't have this, you can't go through. So first big test right here, this checkpoint. What's going to happen, guys? Remember, what was going to be that negative feedback? If we don't have enough, isn't it just going to be that malonyl-CoA? If I see that there, right, made in my fatty acid biosynthesis, if you start to have too much of it, it's going to inhibit the carnitine transporter, preventing fatty acid from getting into the matrix for my oxidation. Okay? Because remember, isn't everything still going to end up as a negative feedback system? Do you ever in the body want to overproduce anything or underproduce anything? That's really what it's all about, right? If anything, even water can be toxic at the end of the day. We never want to get out of that basic homeostasis. Exactly, Eve, I really like that you just said that because you're wasting energy, okay? Excellent job. Good, guys. Negative feedback system are just exactly right, thinking about that equilibrium overall. So let's take a look now at a couple questions here. So we're going to run through these questions together because what I want to show you is kind of a wow moment here. When you look at questions like this on test day, I want you guys to start to think to yourself, why is this an opportunity for me to actually increase my score? Because when I look at this, it's not necessarily as in-depth as the way you once looked at it. Think, if I first read this question, what's the first word that's going to stick out to you? First word got to stick out to you is going to be false. Good. Now we're tying back everything that we learned in our course, still from the same strategy perspective. If I see something that's false, it says which of the following statements regarding NADPH is false? Well, does anyone happen to notice where one answer actually explains why another is true? Which would mean, if we're only looking for one answer choice, I can actually eliminate two answer choices right off the bat here. Yeah, I'm going through a process of elimination for sure. Well, if cells tend to have higher levels of NADPH than NADP+, then theoretically, isn't that why NADPH is used as an electron donor? That would make sense, right? I mean, logically, even if we got to this question, what I always want to think of is worst case scenario. Even if you got to this question and couldn't remember anything, then theoretically I can actually eliminate B and D to begin with. Well, now I'm stuck with A and C. But I remember that the function of NADPH is a reducing agent in the biosynthetic reactions. So if it's a reducing agent in the biosynthetic reactions, what's the next answer choice I can for sure eliminate because it's a really easy process? Boom. Guys, how much easier is that question when we utilize the same strategy from classes, now just looking at the major overview? Is this as bad as you necessarily think? And I know, I would guarantee, all right, if you go back in your diagnostic or your last couple full lengths and you look, sometimes we miss questions like this just because we forget to read the question stem and apply it back with this same concept, okay? Not bad, not bad. Let's go with one more here before we get learning goal 2. Okay, I'm going to give you guys a second to read this, all right? Now it says in beta oxidation of our fatty acids, the fatty acids enters the mitochondrial matrix attached to what? Now, if you see one like this, the question always becomes, will the MCAT sometimes be as straightforward as this? Will the MCAT sometimes be as straightforward as this? Yes, it will. Because if you think about it, yes, I love that you just said that, Tom. This is a pseudo-discrete question. But chances are, if you were to take this and poll all of America taking the MCAT, guess what? Over half of the people are going to get this question wrong because they just simply didn't prep, right? This is where the MCAT is going to reward you for going through and understanding exactly, Rochelle, high-yield important concepts and the pathways that happen. So if you can just visualize this in your head, I almost like to think of it as like a picture. Like if I can see that slide, my fatty acid, the higher concentration, I'm going to go through my mitochondrial matrix eventually. I know that I have to go through the carnitine shuffle. Once that happens, how long does a question like that take you on test day? How long does a question like that take you? Yes, guys, that is what it's all about. Because think about it. Anytime you get into the MCAT, you have to be thinking about yourself, speed and accuracy. I don't want to go so quick that I make silly mistakes. I don't want to go so slow that I never get to those last questions in that passage. Very good. Now, Roberto, I was the same way. I was once a Kaplan student. All right, it worked out. That's why I'm here today. But I actually used to go through and think to myself, oh, no, it's a trick. It's not a trick. Trust yourself. You know the strategy. You know the content. That is really what's going to be the name of the game in the long run. Because there's a lot of easy points out there and easy questions that sometimes we leave on the table because we're not just reading through. So just by looking at that first pathway, remember, we still want to go through and understand our basic pathways, but we also need to make sure that we're tying this back big picture. I love this learning goal because what I think it does is ultimately highlights why are we even learning this and how does it come back. I mean, think, you're going to have to know this from med school, so you might as well know it now. So in learning goal number 2, we're going to discriminate between different energy states and the function of the major hormones in the energy balance. What is the biggest glucose hog in the body? This is why sometimes we get a little bit irritable after studying, right? Sometimes the best trick is just a Gatorade. Yeah, the glucose with the brain, okay? So theoretically, if you start to limit the amount of carbohydrates or just glucose that you can get into your blood, eventually you're going to get into ketosis. So oftentimes when people talk about like bodybuilders or sports physiology or endurance physiology, then what happens is you get into this idea, okay, of what pathways are we utilizing. So you get the glucose for the brain here. Fatty acids and ketone bodies, like we talked about earlier, is pretty much going to be that heart. And then you get the glucose, fatty acids, and ketone bodies. What are we trying to highlight on the bottom here? This is going to be your, exactly, your muscles, right? Skeletal muscle can rely on many sources. So what we'll see here, let's go one step further. You have the liver, we have the adipose tissue, and we can think about the concentration of glucose and fatty acids after we eat a big meal. So this is a big, quick, easy review right before we do a passage to finish up our session tonight. Think about it. What's stored in the liver? What do we like to store in the liver? Think of a big, we're going to spike my insulin, and what we're going to go through is glycogen, of course. So if you get an increase in the concentration of glucose and glycerol, what's going to happen is you're going to go through glycogen storage and my fatty acid synthesis. Because you get that insulin response right after you eat that big pizza, right? Well, now in that pizza, you also get your fatty acids. So if you have that triacylglycerol bound to VLDL, what's going to happen is if you start to increase the concentration of your fatty acid triglycerol, where do we store that? Commonly associated, of course, with your future patients in your adipose tissue, okay? So the idea behind this with your triacylglycerol is you get storage in really 2 spots. So you don't want to forget in the long run why you have the storage in both spots. If anything, you just still have is 2 alternate pathways to bring this through. But I digress. Let's go a little bit further with this. Lipolysis, is the hydrolysis of your triacylglycerol. So what that's going to do is if you have the lower amount of fatty acids, it's literally the same thing as gluconeogenesis. When I don't have enough glucose, I want to break down that glycogen. So if you increase those fatty acids and you get that fatty acid oxidation, exactly, Tom, we need energy. So you're going to go through ketogenesis, gluconeogenesis, and then the glycogen breakdown. And really what that does is it's a response, and now we start to have a little bit more energy. So think about it. If you go through that parasympathetic and sympathetic nervous system, what system are you really activating when you lower your concentration of blood glucose or fatty acids too much? Yes, I love that mnemonic, Tom. because you're in parasympathetic when you rest and digest. And so what ends up happening is if you activate that sympathetic nervous system, think of the caveman running through the jungle. Of course, what you're going to do is be able to utilize that energy. So there's a quick review here on all of the things that you'll see with insulin and glucagon before we do our passage. Now, remember, insulin is released in kind of that absorptive state by our beta cells in the pancreas, and it's dissolved in the blood and it binds. So what I actually like to do here is show you guys, it's just a quick review, but what we want to see is with this review, we're going to jump into a passage, show me the points, right? The idea behind this is you can go through and literally use this slide deck. So what I encourage you to do with your PDF is think of this as on your short list of things to kind of use as a review. What you see when you have insulin is the positive and feedback, and what we're going to relate it back to is our 4 major pathways. So check this out. When you think about increased glucose uptake and acetylcoenzyme A production, where, remember, anything in pink is what? What is anything in pink going to be? We talked about this earlier. What are these enzymes used in? Glucokinase, PFK, pyruvate dehydrogenase, PFK1. Boom goes the MCAT magic dynamite. Now, if I want glycogen production, isn't insulin also going to have a positive feedback on glycogen synthetase? Well, yeah, of course it is. Because what I think of here is I want to then have a negative feedback on my glycogen phosphorase. I want to spike insulin is in response to what? Elevated glucose or lowering of glucose? Think. You guys already know the answers to all of these questions or any of the enzymes that I'm going to bring up. Exactly. If you have elevated glucose, you want that spike in insulin. So even though people will talk about it as bad, right? The humans were not because of obesity and rising epidemic levels of obesity. You don't want to talk about eating sugar and things like that. Isn't it still good that the human body, from a survival standpoint, an evolutionary standpoint, can use sugar as what? Is it a quick or a long onset energy source? It's quick. That's why you have, and now if you go from this fatty acid synthesis and the storage, think about the caveman. He walked all day, right? Brings that big meat, big meal back to camp, right? Stuffs his face to get as much stored energy as possible. That mastodon, well, guess what? I want to increase all those enzymatic levels. I'm going to stop beta oxidation, but I'm also going to go through my VLDL clearance, and what I have then is my triglycerol synthesis. So I'm synthesizing things that are going to store or use energy. Think about it. This is how easy this tends to be. You just have to go back and understand, if I know the things in the pink, I'm storing energy. That's all it is. Now last, but certainly not least here, gluconeogenesis, lipolysis, ketogenesis. What do these 3 things do? What do those 3 things do? We just talked about it. They make energy. All right, watch this. What's the opposite thing of insulin? I don't want to give it away. Let's just think, what's opposite of insulin in the body? Glucagon. What's going to change here if I use glucagon? What's going to change there? Isn't theoretically all the positives going to start to turn to negatives, and all the negatives going to start to turn to positives? Because ultimately what I'm doing is I want to start to have fatty acid release. I want to start to have glycogen use. Look at this. Reduce glycolysis in the liver and increase glucose release. That's exactly what we want, right? I'm going to have that negative feedback system. But now I just stopped glucokinase, PFK2, and pyruvate dehydrogenase. I need to stop because I need to produce a little bit more energy. I'm in my sympathetic nervous system. I want to spike something. I need energy. I need it now. I'm running away from the lion. I need to catch the mastodon. Well, my glycogen catalysis, well, think about it. Is catalysis build up or break down? Does it build up or break down? You betcha. I got to break it down. I need to use that as an energy source, right? I get my glycogen phosphorate, my glycogen synthetase. I want to use glycogen phosphorase. I got to break this stuff down because I need my fatty acid release. And check it out, gluconeogenesis, I'm going to increase. All of these are enzymes that we've seen before. Remember, on our metabolism decks, what are the things in pink? Boom. It's easy stuff to go back and review, guys. This is why it's so cool to come to the channel and start to see what we're trying to show you guys here is the connection All right, we know you're going to have to go back and revise and review some of your enzymes. That's completely okay. But now that we start to understand, hey, if I have my fatty acid concentration rise in the blood and I can use that beta oxidation and go through my ketoneogenesis, I'm starting to get my fatty acid release and oxidation. But think, if you're going through and we're thinking about the body in an evolutionary standpoint, if I have my fatty acid release and my gluconeogenesis, which one is going to give me better bang for my buck in terms of my deposit and withdrawal in my energy systems? Yes, my fatty acids. Absolutely, 100%, you guys got this. So when we go back to how to apply this to the MCAT, we're going to take a look at a passage together to finish this off. But this, guys, is where the MCAT magic comes to play. Okay? So let's check it out. We have exactly a passage and 4 questions left. If we have 10 minutes, should I theoretically be able to go through this passage in 10 minutes and have 4 questions? You betcha. Okay. We've already seen the strategy, right? We've already seen all the things in our classes to know that I can work through a passage, I can use my outlining, and I can start to understand, hey, I want to get into a question. What I want to do is I want to show you guys an inside look. If I were taking this passage right now and doing it on test day, let's try to see, hey, can I just go through, understand what's happening here, but start to use this to my advantage? Because now if I understand the big picture concepts, just a little bit of what's happening here, let's check it out. So we're going to go through and read this together. I want you guys to actively be thinking, as I go through my first paragraph, how could I theoretically outline this to use this to my advantage? Upon first glance, the only thing that I see, if I'm just previewing my passage, is I have 4 different experiments. So what it says is physiologists have performed many experiments using a technique called parabiosis, in which 2 organisms, so let's just say, for example here, I don't know what this is. I've never seen this before. I don't know what parabiosis is. It's okay. The passage is going to give me some information. Now, in which 2 organisms are surgically connected in a way that causes them to share one or more physiological symptoms. And this seems weird to me, but I don't care because I got my background info here. We're good. In the most common form of parabiosis, the circulatory systems are joined so that both organisms have access to anything that enters the bloodstream of either organism, typically by ensuring that approximately 2% of the cardiac output of each animal is directed to the other animal. All right, well, 2% isn't a ton, but I know it's something. So what could be the result of organisms sharing the cardiac output? Probably that there's going to be endocrine hormones transferred between. They can use glucose, fatty acids, something like that, in that nature. All right, but at least we have something there. Okay. Now, parabiosis has been used extensively in studying the digestive system. Consider the following parabiosis experiments. But I do want you guys to think about this when you preview a passage. If I have something like this, what topics come to mind that could be tested in my questions? Because remember, the MCAT's always bringing topics together. What could be tested? I have bio for sure, but if I go into it, I could have the digestive system, I could have hormones, I could have metabolism, of course, because that's what we're covering right now. Yep, your blood, we could get a little bit of physics with the kidney, blood pressure. See, now you guys are thinking like Kaplan students, and ultimately, good test takers, because we're starting to think of how this comes together. Now, this is still going to be an experimental passage, so let's take a look. Experiment 1. Now, as you were going through this, just like I would be, I'll be writing at least the gist down on my scratch work, just like we talked about in our classes. Now it says mouse A, an otherwise normal mouse, is connected parabiotically to mouse B from a strain in which proliferation of the alpha cells of the pancreas is decreased, but not 0. Okay, well, what's the function of the alpha cells in the pancreas? Let's just try to pull that off real quick. Be careful, it's not insulin, it's glucagon. Okay, beta cells is your insulin. Good. So I'm not really, I don't care about this yet. I want to get into the questions before I have to go back. Experiment 2. Mouse C is fed of a diet in which glucose is the only carbohydrate source. Okay, well, mouse D is fed a diet in which fructose is the only carbohydrate source. Still sugar, so it's okay. After 2 weeks on these diets, the 2 mice are connected parabiotically. The diets are continued after the mice recover from the surgery. Whatever. Mouse E is an otherwise normal mouse connected parabiotically to mouse F, which has a type 2 diabetes-like condition but does not require insulin injections. What is type 2 diabetes? Real quick. You got to be thinking here, big picture. Is that insulin is gone, or I'm just not as sensitive to insulin? Exactly, bad receptors. So I still produce insulin overall. So mouse F, insulin production is unaffected. Good. Now, if you have mouse G in experiment 4, has been kept on a starvation diet, given just enough glucose to ensure its survival, because the mouse's brain needs a little glucose. right, for 5 days. Mouse H is an otherwise normal mouse given a normal diet for the same period. After the 5 days the mice are periodically connected. Their pre-surgery diets continue for 48 hours after they're connected. Okay, out of these 4 experiments, which seem to be the most polarized opposites? Because watch, we're going to go through our questions rather quickly here, but you'll notice we can use this to our advantage. Okay, exactly. So let's jump into question 3. In mouse G, approximately 24 hours after the start of the starvation diet, one would expect to find relative to normal mice higher levels of what? Stop and think here. Which experiment was mouse G talked about in? Four. So is mouse G technically the mouse being starved? Okay, if mouse G is being starved, then I don't have a glucose source. Now wait a sec. Mouse G, in the fasting state, what process tends to increase while all of my others would tend to decrease since I don't have any energy source available to me? Think about it. Ah, yes, guys, that's excellent. Because think, if I don't have a glucose source, what hormones may be present in mouse G's blood due to starvation? I need to go through glucagon, right? What's going to end up happening is gluconeogenesis. Now, one of the things that people commonly confuse, would we be going through fatty acid synthesis if you didn't have glucose and you started to go through that process? No, we want energy. So aren't we breaking down our fatty acids in order to produce a little bit more energy here? It takes a little bit longer. It depends on the animal. So for mouse, it's actually a little bit shorter. In humans, it usually takes up to 72 hours. It depends upon your metabolism, actually. You ever look at like natural bodybuilders and stuff like that, use the same idea, or endurance athletes talk about being fat adapted? That's part of the process. But kind of outside the scope of the MCAT, you don't have to think about that. Just think about what they're asking you. Now, question 4. Before the surgery in experiment 3. Now, really quick, if you had to think about experiment 3, what was really being talked about there? If I had to write something down for my outline, it would probably just be, I'd probably just write down diabetes, right? That type 2, I'm insulin insensitive. Good, Andrew, that's exactly right. So it said mouse E weighed 25 grams while mouse F weighed 50 grams. All right, that's almost double. So as soon as I'm going through that question, I'm thinking, all right, that's got to be a clue as to something, right? Well, which of the following results would be most reasonable a week after the surgery? Well, a week after the surgery, now wait a second. If you're type 2 diabetic and mouse E weighed 25 grams, what characterizes diabetes is going to be that insulin resistance. So if that mouse E was type 2 diabetic, right? Or excuse me, mouse F was type 2 diabetic. Sorry, I just misspoke, right? And mouse E was otherwise normal. Is there any reason for mouse E to have gained weight? Is there any reason for mouse E to have gained weight? No, because the mouse E is acting normally. So if I go through that process and the mouse E is still going to be normal, what 2 answer choices can I eliminate? B and D. Well, now that I look at this, if you have mouse F, who was the diabetic, then wouldn't mouse F, there's no way mouse F could have lost weight, right? Mouse F would have had to have been obese. There's no reason to assume, exactly, just like Eli was saying, that it would suddenly double its weight for any sort of reason if it was normal. Excellent job, guys. Yeah, so Eli makes a good point, is the cardiac output is another thing to think about. It's only 2%, so it's going to be a drastic increase. Use your common knowledge and what you know. Now, lastly here, you look at question number 5. Look how quickly this question is. Let me use this as an example. After the surgery, hepatocytes in mouse B would likely exhibit increased activity of what? Well, if I think about the increased activity, I know that I have a Roman numeral question, right? Excuse me. So what happens is this surgery should increase blood levels of glucagon, which would increase things activated by glucagon. Well, that's it. This question tends to try to be more confusing than it really is. But if it wasn't present in mouse B before prabiosis, glucagon's present now. Now, in this question, even though it's a Roman numeral question, you do not have to go into as in-depth as you want. Would glycogen phosphorylase be increased if you increased your glucagon, based upon our chart that we looked at previously? Yes, of course. Now, I wanted to start with 1 because it showed up most often in my Roman numeral questions. Would hormone-sensitive lipase also increase if you had glucagon? Of course it would. And then if you look, what about PEPCK? Now, that's the one that sometimes people forget. That's involved in gluconeogenesis. So I know it's got to be answer choice. I want to show you guys one last question here. We'll finish up for the day, taking away that this is not as intimidating a topic as originally thought. Now, question 6, 3 enzymes in glycolysis and 3 enzymes in gluconeogenesis are unique to each pathway. Which of the following is true of all of those enzymes? Well, think about it. If you go back, and I want you guys to look at this for homework, I want you guys to remember that what do all the enzymes unique to glycolysis and gluconeogenesis have in common? And this is the one that sometimes people forget. They catalyze irreversible reactions. So if they're irreversible reactions, this question is super straightforward. The only thing that you need to look for is you're looking for something that's going to catalyze spontaneously under physiological conditions. All right, Andrew, close. We're going to leave you guys with 6 for homework, but what was the main takeaway, guys? What do you now think, if you look back and we look at all of this metabolism, I want you guys to remember here, is this as bad as it initially seemed when we started tonight's session? I know it's an intimidating topic, but is it really as bad? I know, Tom, it's not as bad. The one thing that I'm going to highly encourage to make sure you guys do, just like we talked about, everything in pink were those enzymes. So you have to think to yourself, when I'm going back, okay, exactly, that circular process, if you can think about the cyclic going through, you have to understand the inputs, the outputs, but most importantly, the connections. You guys are one step closer to that higher MCAT score. And as we leave every session on the channel, I just want you guys to remember, success is when preparation meets opportunity. A little bit of prep and metabolism is going to go a long way. All right? So, guys, excellent job today with a really tough topic. It was another great channel session. I got Kristen Russell coming up next. So I wanted you guys to stick around, have a great day, and I hope to see you guys back here on the channel very soon. All right? I'm going to go grab a cheeseburger. I'm hoping for that fat metabolism. See you guys. Have a good night.